So we have an episode about spindle neurons, sometimes called von Economo neurons, and these are very relevant to the autistic phenotype. So these are spindle, sometimes called von Economo or VEN, von Economo neurons. Von Economo kind of discovered these in the 1920s. Before that, though, in medical literature and medical textbooks, there were some mentions of it around the late 19th century, 1880s, and so forth. I think there were some mentions of it, how there's a certain type of neuron that's shaped a little bit different. We'll draw them out shortly, but these are called spindle neurons because of the shape. More recently, over the last 20 years, a researcher named John Allman has studied these quite extensively. He has a lot of good research on the spindle neurons. And these are very fascinating because they are only found in a few type of creatures, very socially active type of creatures. So humans have them, larger primates like orangutans, gorillas, bonobos, a few cetaceans like whales and dolphins have them, elephants. So there's only like 10 or 12 different species that these are discovered in. And humans have them in primarily two different regions, two regions that we've discussed quite uh, in detail: the anterior cingulate cortex, or the ACC, and the insula, specifically the frontal or the anterior insula. So these spindle neurons are they're provided to have rapid transmission for social context is what they're doing. They are very larger than normal neurons, and they only act mainly on the different types of monoamines, especially dopamine and serotonin, and a specific cell peptide that we've never discussed, I don't believe, and that's neuropeptide Y. It's not really discussed quite quite frankly. And some of these things are well, this is a peptide, so as if you remember the oxytocin and vasopressin episode, peptides are just a collection of proteins, amino acids. So oxytocin and vasopressin are pretty easy because there's only 9, and they share the 7 of the 9 peptides. But neuropeptide Y has 36 protein bonds or chains of proteins, and the neuropeptide Y is heavily involved with fear buffering. So it kind of makes sense if you're navigating a social world. And whenever I'm talking about social world, it's not just necessarily communication or hanging out with others and so forth. It's not really what this is about. That's more of a specific type of social environment. This is just interacting with anything from the environment. Humans are very social creatures, so we're constantly being entangled with many different types of things in the environment. Other things with the neuropeptide Y is regulating things like food intake and metabolism and autonomic functions, things like breathing and heart rate. So, it's a peptide, so they have a pretty diverse type of functioning. The peptides do, similar to the oxytocin and vasopressin. So these two brain regions right here are very critical, and that's why I just listed them out before we got started so we can go over the roles of them. So the ACC, the anterior cingulate cortex, which is here, it's it's part of the medial prefrontal cortex. It's commonly just highlighted as a medial prefrontal cortex nuclei or subdivision. It has massive roles, and we've covered these roles in several different episodes, but conflict monitoring and error detection, evaluating risk and reward. So potential choices and outcomes is what this is kind of doing. We talk a lot about internal calculators, ACC is a huge internal calculator for these types of things. Goal-directed type of behaviors. So it's in the medial part. So typically with things like the prefrontal cortex and the dorsal striatum, the more medial you are, the more in the middle or inside, deep inside the brain, is more reflective of goal type behaviors. And then the habits kind of branch off laterally, okay, as we evolve as species, different types of species. It's goal directed on the inside, and then habit forming will come later because that requires a lot of repetition. So it grows outward, is a good way of thinking about that. In mice and rat studies, there are good goal directed versus habit formation. And you can do things like outcome evaluation to kind of disassociate the behavior because you can't ask the rat or mouse. They can't tell you. Controlling autonomic functions as well and physiology. And we often say right here, adaptive responses. When we're responding well in the environment, that's called adaptive responses. And with autism, you can think about just an overarching way of describing it as a lack of adaptive responses because there's not a lot of skill set. There's not a lot of social databases and skills to pull from as we navigate the world. Second is the insula. The insula is a very fascinating region because it has privilege. It's very privileged to having information coming in about the brain, what's going on inside the brain, the thoughts and the feelings and so forth, the different brain waves, and the body. So it's getting information from the physiology. My heart rate's okay. I'm okay right now. My breathing, the depth and rate, I'm okay right now. But what if you're not? What if I'm just sitting here, or you're just sitting around, not doing much physical activity, and your heart rate's jumping, and your breathing's kind of, the rate and depth is kind of off. You might register, oh, something's not right here. Well, that's feeding up to the insula to inform the rest of the brain, something's not right here. And the insula is also getting information from the environment. So 3 main data sources to pull from: the brain state, the body state, and the environment. How is the living organism incorporating all 3 of those? So the insula is a very fascinating little region here. And it's right here. This is the frontal part. And then the posterior part kind of expands this way. Now the insula is very lateral, okay, and especially in comparison to the ACC and all of these other subcortical areas that we'll go over. It's very lateral. It's more likely that it came on board later in the evolution. This is also known as a gatekeeper in task switching. So one thing that's fascinating about the cingulate cortex is, and it doesn't matter if it's the anterior, the posterior, the subgenual, whatever. The cingulate, let's imagine these are white fiber tracks, nerve fibers, or so forth. Okay? Remember, we've talked about things like the fasciculi and things. These nerve fibers, myelination and so forth, they connect brain regions, distal brain regions. We'll go over the mini columns as well because spindle neurons are heavy in layer 5B. But the cingulate, and I learned this from Nolan Williams, MD, PhD, just passed away, very, very, very brilliant researcher, huge with investigating depression and things like that. So hopefully his soul is resting in peace. But the cingulate wraps around white matter tracks, like this. Okay. So it's just wrapping around white matter tracks. And that gives the cingulate a lot of access, a lot of fast acting access as well. So the insula and cingulate cortex, it makes sense why it's really involved in these kind of fast acting, rapid transmission type of social bonding type of neuron. Okay, so if you remember, we've covered a network, the so-called Salience network. These two regions are essentially the main regions, the hubs of the Salience network. So this is a big thing. The Salience network allows us to divide our attention. What are we going to pay attention to? What is most important to the living organism? Okay. So you have the ACC and the interior insula. And then you have all of these subcortical areas. Things like the ventral striatum, which is mostly the nucleus accumbens. Um, the nucleus accumbens is a very large region, to be frank. The thalamus, of course, the hippocampus, the substantia nigra compacta. Remember the episode with Parkinson's and the basal ganglia? The substantia nigra compacta is dopamine. The reticulata, which is it's like a Nike check almost. And the check part, the part that comes up, is the reticulata. And then also the VTA, the ventral tegmental area. This is dopamine. So these two are dopamine areas. And then the amygdala and the hypothalamus here. So the Salience network, these are mostly the hubs, and these subcortical areas are kind of nodes, what's called nodes. So collectively, these are kind of all working together to help the living organism send our spotlight of attention to whatever we find most salient, Salience network. And then also the spindle neuron, in 2008, there's small clusters of the spindle neurons found in the dorsolateral prefrontal cortex, just like clusters of two. So they're very, very sparse, rare. With the interior cingulate cortex and the insula, so we have approximately 100,000 to 200,000 spindle neurons in the central nervous system. These are just neurons, okay? Now, overall, the human body has roughly 85, 88 billion neurons in the central nervous system. So there's not a whole lot of these spindle neurons in comparison to overall neuron. What I want to really say about here, let's just go ahead and erase this. Because I've talked a little bit about several different times the paper from 2024 about the Salience network with 6-week-old human beings, human infants, and the Salience network. This was done at UCLA, Sang et al. And Lucinda Uden is on this paper. Uddin. Lucinda Uden kind of really captured the role of the insula. And about 2008, 2010, and so forth was a really hot year for the insula, or years. It still is. But on 6 weeks, this study on 6 weeks though, they used fMRI during non-activity, so it's almost like a sleep state on humans though. And they found that children that had a high likelihood of having autism. So they had autism as their siblings already had autism. So they would hook fMRI up to these human babies and just check their connections essentially. And the high likelihood found that the Salience network had a strong connection to this area right here. The sensory motor area, which is right here. A lot of our not adaptive responses, but a lot of our automatic responses will live here versus here. We like to send a lot more reflexive things, habitual things up to here. And then it can just feed down into the dorsal striatum and then kind of sort the basal ganglia for movements and actions. So they found 6 weeks old a stronger connection to the sensory motor area. And if you think about autism and kind of the rigid thinkingness and lack of cognitive flexibility, right here, versus the typical likelihood, the other children, 6 weeks old, they had a stronger connection with areas here. It's a big finding, but nobody does anything with it. It's just kind of lost in the academic literature. Nobody does anything with this stuff. And what's fascinating here is, it's UCLA. It's like an ivory tower. They followed these kids up that, because they were only 6 weeks old, they followed them up to like years 2 and 3. And they could confirm, like, the ones that had a stronger connection to here, definitely indeed, autistic phenotype. Nobody does anything with this stuff, though. So let's draw out the spindle neurons versus things like the pyramidal neurons, okay? So, pyramidal neurons, they have this pyramid, okay? And then you have things like these dendrites and axons falling off of it. And like so. You get the good idea here. It's more like a typical neuron, especially the spindle or the pyramidal. The spindle neuron, the cell body is like that. It's more of like this. That's why it's a spindle neuron. And it's bipolar. So it looks a lot like this. So here's a big thing too. It's much larger. And this allows for that rapid transmission. So the ones in the ACC are actually found to be a little bit larger, even. The ones in the ACC, they can extend down quite a bit. So it looks more like that. Now, if you remember the layers of the mini columns, we had all of these type of cells here. And then layer 4 has the stellate cells, so forth. And then down here, we have more of these and here's where you can find some of these spindle neurons. So they can be like much larger. And so forth. But they're very rare. But they're predominantly in layer 5 right here. Now, not covered in the mini columns little video is this layer down here, directly below these mini columns. Remember, would be considered a mini column, and then we would have very many different mini columns, and it would eventually become a micro column and so forth. This layer, right below layer 6B, 6A and 6B, is white matter. This is where a lot of distal connections will happen. So these spindle neurons, remember, inputs come here to layer 4 from the thalamus, and then it goes to 2 and 3 because this is where the subjective experiences of the living organisms essentially live, is layer 3. Is the glare all right? And then it will shoot down to 5. This larger spindle neuron will allow this information to be received and transmitted down to these white matter tracks faster. And that way we can respond faster to the environment. This is not so much a social thing, like I'm going to go hang out with friends. This is a survival thing. This is an ability to get information from the environment fast so you can make decisions. This is space-time. Again, we're always talking about space-time. And these different spindle neurons are very rare. And they're regarding socialness. And autistics have kind of a complicated number with the spindle neurons. At first, in the first 4 years of life, it's kind of off. And then later on in life as well. It's just post-mortem studies will hit this. So there's a lack of kind of these types of cells. And, if you think about the comment about the adaptive responses from earlier, it makes socialness, the outside world, I should say, complicated. And because of this region right here, the insula, the outside world, and the Salience network, our attention is going to be inward to the self. Spindle neurons.